Hi students, welcome back to another session. Today we will be continuing the same topic, recombinant DNA technology. Do you remember the previous class what we discussed? So just a recap, recombinant DNA. Recombinant DNA we have learned the first recombinant DNA. Who developed the first recombinant DNA? Stanley Cohen and Herbert Boyer. Then we learned about the tools required for the production of recombinant DNA, or in the recombinant DNA technology, which are the tools required. Okay, in that we started learning about the restriction enzymes. Restriction enzymes belongs to the class nuclease, right? And the nucleases, what are the different types of nucleases? Restriction endonuclease there and the exonuclease there. Then we learned about the nomenclature of restriction enzyme. Nomenclature. What is nomenclature? The naming of the restriction enzyme. There we learned one example, EcoR1. EcoR1, from which bacteria this restriction enzyme developed, that is from Escherichia coli, right? Then we started learning the features of the restriction enzymes. There we learned restriction site or the palindromic sequence. Palindromic sequence. What are palindromic sequences? And then restriction enzymes, they recognize the sequence that is called the palindromic sequence. Okay, and one example of the palindromic sequence, EcoR1, they recognize the palindromic sequence GAAT-TC. Okay, GAAT-TC, this is the palindromic sequence. So here in the first strand, 5' to 3' end, this will be the sequence, and the other strand. Okay, so this is one strand, the other strand, 3' to 5' end, it will be GAATTC. So in one strand from left to right, the other strand right to left, it is same only. That's what the palindromic sequence. Till here we learned. Coming to the another feature of restriction enzyme, they produce two types of ends. Okay, one is called a sticky end, the other one is called a blunt end. EcoR1 is an example of the restriction enzyme. They produce sticky ends. Sticky ends, what is that sticky end? For example, I can say EcoR1, what is the recognition site? GAAT TC, right? The other strand also GAATTC. Okay, and the restriction, this EcoR1, they will cut the phosphodiester bond between the guanine and adenine. Always remember, EcoR1, they break the phosphodiester bond between the guanine and adenine. Okay, and here also guanine and adenine in between they break. So we'll get the overhanging, okay, single stranded hanging strand we will get. That's what the sticky end. A single stranded overhangs created when the restriction enzyme, they cut in a staggered way they will cut. Okay, not like a straight cut. Here one side, here another side they cut. So we'll get a staggered cut which result in the formation of sticky ends in some restriction enzymes. So that will facilitate the easy ligation. Easy joining is possible when there is a sticky end, right? And high efficiency in cloning. If the ligation is proper, then the cloning efficiency also will be high. And the most commonly used in research is the restriction enzymes which cut the sticky ends. There are certain restriction enzymes, they cut the blunt end also. Okay, so blunt end, it is very difficult to ligate. Blunt end means like, for example, GAAP. Okay, GAAP like this. The cut will be like this. Okay, so only one side the cut, straight cut, which causes the blunt end. That is very difficult to ligate with the vectors and the what is that the whole gene of interest. Okay, the gene of interest and the vector joining is very difficult in the case of blunt end. So in RDNA technology, most of the scientists, they are using the restriction enzymes having the sticky ends. Okay, so that's all the features of the restriction enzyme. Now let's discuss the mechanism of action of restriction endonuclease. Okay, so the mechanism of action of restriction endonuclease in the NCERT, there is a set of diagrams there, sequence of diagrams showing the mechanism. You have to learn. We will discuss that part. So in the, suppose in the vector DNA, okay, we are drawing the vector DNA. In the vector DNA, and the another one is the foreign DNA. Okay, so foreign DNA is also called a alien DNA. Okay, so alien DNA is there, vector DNA. In the vector DNA and alien DNA, we need to cut the DNA strand. Okay, and with the help of what? With the help of what? We can cut the DNA strand with the help of molecular scissors called a restriction enzyme. Here we are using the same restriction enzyme to cut both the vector DNA and the foreign DNA. This is the vector DNA and this is the foreign DNA or alien DNA. Okay, alien DNA is there and the vector DNA. So we know that we are using the same restriction enzyme EcoR1 for cutting the DNA part. So first of all, the EcoR1, they will check the palindromic sequence. What is a palindromic sequence? GAATTC. This is the palindromic sequence. Here also GAATTC. Okay, and alien DNA also should have the same palindromic sequence. GAATTC, GAATTC. Right? And so first of all, in the mechanism, very first step is restriction enzyme. They inspect the DNA. Inspect the DNA and they will find out the palindromic sequence. Palindromic sequence they will recognize. After that, they'll cut the specific sugar phosphate bond between the guanine and adenine. So sugar phosphate backbone, there are phosphodiester bond that will be cut by the restriction enzyme EcoR1 specifically in between the guanine and adenine. Here also between the guanine and adenine. Here also between guanine and adenine. Here between guanine and adenine they cut. So after the action of the restriction enzyme, we will get the sticky end. Okay, sticky end, one side only the guanine present, the other side you can see cytosine CTTAA. There is no G because here only they cut. The alien DNA also, sorry. So AATTC and here only G is there. Okay, then the rest of the part like this. So vector DNA, alien DNA, and we need to join this alien DNA into the vector DNA. Okay, so this one, the joining is with the help of which enzyme? So cutting is with the help of the molecular scissors called a restriction enzyme. After that, with the joining help of which enzyme? That is DNA ligase. DNA ligase is used for joining the fragments. Okay, so here the DNA strand and the vector DNSG is there. Here C double T A A. This much is there. Now think about the alien DNA. Okay, alien DNA part also we will be joining here. A T T C. And in this here also G is there. This part G is there. Okay. So now this DNA, this become a completed DNA, and this DNA is called a recombinant DNA. This recombinant DNA can be inserted into the host body, host cell. And from the host cell, along with its multiplication, we will get copies of the recombinant DNA. That's what the mechanism of or the steps involved in the recombinant DNA. First of all, we have to isolate the DNA. Okay, so both the plasmid vector DNA and foreign DNA containing desired genes are extracted from the respective sources. Right, and cutting with the EcoR1. Restriction enzyme EcoR1 we are using for the cutting. And we will get the sticky ends. Here you can see the plasmid. In the plasmid, a particular part cut, and then the foreign DNA's part also. Then it is joining with the help of ligation of the DNA fragments with the help of which enzyme? DNA ligase. Okay. Then introduction into the host cell. Host cell, this recombinant DNA will be introduced, and then amplification. Okay, copying of the DNA. That's what the mechanism. So remember all the steps involved in the mechanism of the production of recombinant DNA. Right. So first tool is over, that is restriction enzyme. Moving on to the next tool, that is DNA, the vectors. Okay, DNA carriers called vectors. In health and diseases also we learned about vectors. They are the carrier of disease causing microbes. Here in recombinant DNA technology, vectors are the carriers of DNA. Usually our DNA technology, they are using either plasmid or bacteriophage as a vector. We know what are plasmids. They are present in the bacterial cell, and they are small circular extra chromosomal DNA. Remember, they are the small extra chromosomal DNA, and they can replicate itself. Autonomously, they can replicate. Okay, so that's what they replicate independently of the along with the main chromosomes. So this plasmids are used as the vectors in DNA technology, and they have some specific characters also there. For example, antibiotic resistant gene is there in some bacterial plasmids. Another vector is bacteriophage. We know the bacteriophage, they are the virus which consist of a protein coat. There is a protein coat, like this protein coat, and the DNA is there. Okay, that is the bacteriophage. So bacteriophages are a virus which infect the bacteria. So when this bacteriophage enters into the cell, they increase in the number. So their copy number is more. Copy number of bacteriophages more as compared to the plasmid. There is a board exam question. When you compare the two vectors, plasmids and bacteriophages, which is the most efficient one, and give a reason. The efficient one is bacteriophage because the number of copies or copy number of bacteriophages are more as compared to plasmids. Okay. Yeah, so let's discuss the essential features of a vector. A vector should consist of a origin of replication. It is called a oricite. A vector should contain a particular place where the origin of replication start, and that is called a oricite. And the vector should consist of another important feature, that is selectable marker. We are using markers to mark something, right? In RDNA technology, the markers are generally the genes. Genes used as a marker to identify whether the particular cell is the transformed cell. We are using that transformants. Okay, so selectable markers are used to find out the transformants from non-transformants. So what are transformants and non-transformants? So transformants are the host cell which consist of the plasmid, and the plasmid consists of the specific alien DNA. Alien DNA carrying plasmid is there, then we can call transformant. If the bacterial cell is there, it does not accept the plasmid which consists of alien DNA, then we can call this bacteria as non-transformants. So in the Petri dish, there are transformants will be there, non-transformants will be there. Okay, so we need to separate the transformants from the non-transformants by using selectable markers. So selectable markers are generally antibiotic genes. Genes which are resistant to antibiotics. For example, the genes which resist the ampicillin we can call AMP-R. The genes which resist the antibiotics tetracycline, TET-R. Okay, so this is another feature. Then the third feature of vector is it should consist of at least one cloning site. Cloning site are the specific recognition sequence for the restriction enzyme. Earlier we discussed a restriction enzyme. They recognize certain sequences, the palindromic sequences. Okay, so in the plasmid, at least there should be one recognition site at least should be there. Then only we are considered as a vector in RDNA technology. So in addition to those three important features, the size of the vector should be small. And there is one more site, it is called a ROP site. Can anybody say what is the use of the ROP site? Yes, ROP site is the site which carries certain genes which code for the proteins that are essential for the DNA replication. For the DNA replication, there are certain proteins and enzymes required that is produced from a specific site called a ROP site. So these are the fundamental or the special features of vectors. Remember at least three features. Okay, ori site should be their selectable marker and a cloning site. So we will learn one artificial vector that is called a PBR 322. So all the competitive exams, your board exams, based on this diagram, a lot of questions came. Okay, so you can expect this time also questions from this topic, PBR 322. So what is PBR 322? It is the first artificial vector developed by two scientists. Okay, so their name Oliver and Rodriguez. Oliver and Rodriguez developed the first artificial vector and they are called a PBR 322. P for the plasmid. Okay, this vector is a plasmid. Just observe the diagram of this PBR 322. What and all there you can see there is an ori site, right? So ori site or origin of replication, this is the specific sequence of the plasmid in which what is starting? The replication of DNA. Replication of DNA starts at a specific point called a ori site. Next to the ori site you can see ROP site. What is the use of ROP site? Production of certain proteins which are essential for the replication of DNA. Now coming to the certain other sites, you can see PST1, PVU1, EcoR1, CLA1, HIND3, BAMH1, SAL1 and PVU2. 1, 2, 3, 4, 5, 6, 7, 8. Those 8 sites are called restriction sites or the cloning site. Remember, cloning sites in PBR3 PBR 322, 8 cloning sites are there. What are the 8 cloning sites? PST1, PVU1, EcoR1, CLA1, HIND3. You need to learn all those parts. HIND3, BAMH1, then SAL1, then PVU2. Okay, 8 cloning sites are there. Now coming to that internally, you can see the pink color structure. This is the one of the selectable marker called ampicillin resistant selectable marker. Okay, selectable markers present in PBR 322. One is ampicillin resistant gene is there and another one is the tetracycline resistant gene. So when you check the diagram, you can see in the ampicillin resistant gene here there are 2 cloning sites there, PST1 and PVU1. Coming to the second one, tetracycline resistant gene, there you can see another 2 cloning sites, BAMH1 and SAL1. Okay, so PBR 322, remember all those parts. What are they? There is an ori site, ROP site, cloning sites there, and ampicillin resistant gene. Okay, so they will ask which are the which are the selectable markers present in the PBR 322. What are they? Ampicillin resistant gene and tetracycline resistant gene. Okay, and what is the use of selectable marker? Selectable marker is used for finding out the transformants from the non-transformants. Okay, so there are certain terms you have to learn like a transformants, non-transformants, there are recombinants and non-recombinants. Can anybody say what is the meaning of recombinants and non-recombinants? Yes. See, the plasmid which carry the alien DNA, then we can call as the recombinant DNA. The plasmid which does not accept the alien DNA or the foreign DNA, then we can call as the non-recombinants. Okay, so the coming part we will discuss again the selectable markers used in that. Remember these four terms: recombinants, non-recombinants related to the plasmid, the DNA level. When these non-recombinants or the recombinants enter into the organisms, then we are using the cell transformant and non-transformants. Okay, this much is clear, all of you. Okay, so that's all today's class. If you have any doubt, you can ask related to till here. The properties of restriction enzymes, the features of cloning vector, PBR 322, the different parts of the PBR 322. So now this is the discussion or doubt session part. You can ask your doubt now. I hope there is no doubt. Okay. So we will wind up today's class. Thank you, all of you.